Hello students, this is Dr. Ravi Sani. Today we are going to discuss about the blood groups. Now come to the various types of grouping of blood has been done. There are so many types of blood grouping. The commonly used blood groupings are the ABO and the RH system. They are widely used all over the world. Now come to the ABO grouping. ABO grouping is based on the presence of two surface antigens, or we can call them agglutinogens. They are chemicals that can induce immune responses on the RBCs, namely antigen A and antigen B. So ABO grouping, it is based on the presence of antigens. Basically, they are surface antigens. We could call them agglutinogens A and B antigens. Similarly, the plasma of different individuals contains two natural antibodies. Antibodies, these are the proteins which are produced in response to antigens. In plasma, antibodies are present. The distribution of antigens and antibodies in the four groups of blood. There are four groups of blood on the basis of antigens A and B. The first group is A, second is B, third is AB, and fourth one is the O. In this table, these are the four blood groups A, B, AB, and O. These are the respective antigens which are present in the respective blood groupings. In case of A blood group, the antigens are present which are A. In case of B blood grouping, antigen B is present. In case of AB blood grouping, both antigens are present. In case of blood group O, there is an absence of any antigen. Now come to the antibodies which are present in plasma. In case of A blood grouping, the antibody that is present in plasma will be or would be anti-B, because if same group of antibody will be present, it might lead to clumping or agglutination reaction. In case of B blood group, the plasma contains anti-A antibody. In case of blood group AB, there will be no antibodies in plasma. In case of O blood group, both the antibodies anti-A and anti-B will be present, because there are no antigens in case of O blood grouping, so there will be no reaction. These are the donor groups: A and O, B and O, AB, A and B, and O. In case of O blood group, there is only O blood group for donors. In this image, the further explanation of these presence or absence of antigens could be seen. Here is the antigen A, here is the antigen B. In case of AB blood grouping, both the antigens are present. In case of blood group O, no antigens are present. The respective antibodies will be absent in case of O blood grouping, and there will be present both the antibodies A and B in case of O blood group. RH grouping. The RH antigen, similar to one present in rhesus monkey, was first observed in RBCs of majority. Almost 80% uh, people people contain RH antigen, so they are under the category of RH positive. The remaining percent could be put into the category of RH negative. Now come to the various types of RH antigens: C, D, E, CD, and E. There could be some other antigens also, but the main antigen, that is antigen D. So on the basis of this particular antigen, which is more uh, prominently responsible for the RH antigen incompatibility, that's why mainly Rh antigen could also be called D antigen. Other than D, there are some other antigens also. Basically, uh, there's a whole range of Rh antigens. These are some few antigens which are pointed here, and among these few, antigen D specifically could also be represented as Rh antigen. The type D antigen is widely prevalent in the population. That's why it is considers more antigenic than the other Rh antigens. Anyone who has this type of antigen is said to be Rh positive, whereas the other groups who doesn't have such kind of antigen would be categorized into Rh negative. If Rh negative person is exposed to Rh positive blood, it will form a specific antibodies against the Rh antigen. Therefore, Rh group should also be matched before transfusions. There is specific disease which occurs due to this specific condition. If Rh person is exposed to Rh positive blood, which is called as erythroblastosis fetalis, we'll discuss. We have studied the antigen. On the basis of antigen, we have divided the blood grouping A, B, AB, and O. These are the naturally occurring antibodies in the plasma of subjects who lack the corresponding antigen, even if they have not been transfused or been pregnant. These are the naturally occurring antibodies. The anti-A and anti-B are the naturally occurring antibodies which are present in the respective subject who doesn't have the relevant antigens.